Hello, my name is Steve Murphy. I'm the AssayMAP product owner, which means I've been involved in developing this product for a very long time and understand the design inputs and the uses of it. So I should be able to answer questions that are raised. So what I'm going to do today is go over an overview of the AssayMAP Bravo and the expanded capabilities we have with the new large capacity cartridges. So here's the overview. So I'll give you a quick overview of the system and then talk about how we balance flexibility and ease of use, and then focus on the large capacity cartridges. So I'm going to focus on sample prep, and the sample prep is going to be automated sample prep using the AssayMAP Bravo. But I want to be clear that when we're developing the sample prep applications, we do it in the context of a full workflow. So we know that we have to be cognizant of the samples coming into the sample prep process and our handoffs to the separation analytical instruments, because ultimately we're trying to get the best solution so you have the best data to make good decisions for moving your projects forward. So this is the AssayMAP Bravo platform. It's composed of three parts. The first part is the AssayMAP Bravo itself, which is the Bravo liquid handler. And what makes it an AssayMAP Bravo is the AssayMAP Bravo head, which contains 96 probe syringes arrayed in an SBS format. These probe syringes are controlled by the Bravo, so they have very precise flow control. So in essence, what you have is 96 syringe pumps that are very precisely controlled. Now, the second component of the AssayMAP Bravo system is the AssayMAP cartridges. These are packed bed cartridges. So these cartridges have cartridge beds in them that are chromatography columns. When you combine the precise flow rate of the AssayMAP Bravo syringe pumps with the columns in the cartridges, what you have is a chromatography system that can process 96 samples in parallel. But it's not limited to 96. You can do anything from 1 to 96 samples. The last component of this platform is the user interface, the software, which is critical because really powerful technology that doesn't have a good user interface is not very useful. So we spend a lot of time developing applications with their focus being use by bench scientists, so that you can quickly be up and running and access the power of automation without having to go through all the programming and learning software. So if we take each of these pieces and individually look at them, again, we have the AssayMAP Bravo head shown here. Now, if we look at the syringes that are in the AssayMAP Bravo head, one thing that I wanted to point out about the syringes is that the plunger here is shaped to fit perfectly into the barrel of the syringe. This leads to very little or no dead volume, which makes washout and very effective. If we look at the cartridges associated with the probe, so first of all, the cartridge here beside the US quarter gives you a sense of the size of the cartridge. And the most important thing is seeing that the end of these probes sit in the cartridges right on top of this packed bed. And importantly, there is no air here, it's just liquid. So there's a liquid-liquid interaction. So you have highly precise syringe pumps that can deliver very well-controlled flow rates over a packed bed column. So you really truly have chromatography and all the benefits that come with it. If we look at the cartridges, as everyone who's tried to pack cartridges on their own before, or columns on their own before, you know that the quality of the column is really critical for getting good chromatography. So this cartridge has a top and a bottom frit with a packed bed in between. To ensure that we have high quality and consistent cartridges, we have an automated packing system. This packing system does 100% inspection, so every cartridge is inspected. And the way we do that is to flow liquid across the cartridge bed. Now, the pressure and measure the pressure drop. the pressure drop is indicative of the amount of resin and the packing in the bed. So we can very tightly control the quality of the cartridges and make sure they're very consistently packed. In addition, the automated packing system has machine vision, which will look for any abnormalities. And in either case, if there's a problem, the cartridges are automatically thrown away. Now, these cartridges can be packed with a variety of resins, and here is showing some of the resins with their surface chemistry, and I'll get back to more details about this in a later slide. Lastly is a user interface. Like I said, it's really important that we make a simple user interface. So here's an example of affinity purification. What you see here at the deck layout is showing you where you put your solutions on the deck. For every deck position, you have a pull-down menu, so you can choose a different type of labware depending on your experimental design. And then importantly, here are the application settings on the left side of the screen. And they're set up from the way the robot runs from top to bottom, but they're also very clearly designed for bench scientists. So you have a prime, an equilibrate, a wash step, a second wash step, and an elution. So very intuitive, very simple. Each of these steps can be toggled on or off to make it flexible. So you can, for instance, if you want to do 2 wash steps, you can do 2. If you only want to do 1, you toggle the second wash step off and it doesn't happen. You can control the volume, the flow rate, and the number of washes after each step. And what you'll notice is that these have values in them. So we've already optimized and shown the volumes that we feel will give you really excellent results. But if you wish, you can change these if you have a certain situation where you feel like you need to optimize these conditions. Most obviously, you would change the sample volume because that's going to change from experiment to experiment. So in summary, we have a system where we have precisely controlled syringe probes, which interface with very nicely packed columns with no air gaps. So it's a liquid-liquid interface. That gives you all the components that you need to do chromatography, and you gain all the benefits of doing your sample prep with chromatography, such as high reproducibility, quantitative binding and elution, highly efficient washing because it's a unidirectional liquid flow, very small elution volumes, which really can decrease your time to results, especially in the context of the entire workflow, and you can process 1 to 96 samples. So now if we talk about what if we built into the system to increase its flexibility and ease of use. So there are 2 diametrically opposed design inputs here. One is that you would really like to have as much flexibility as possible, but with maximum flexibility also comes maximum complexity. So the ultimate would be you write your own scripts and everything else, but that takes a lot of time and a lot of energy. On the other side of the spectrum is ultimate ease of use, which is simply a black box where you click go. We need to be somewhere in the middle because we're dealing with samples, primarily protein samples, which require some level of flexibility. So how do we design in the kind of flexibility you need to address the kind of samples that we deal with, yet is easy to use? So there's a number of ways we've done that. The first is that we have 2 different size cartridges that are available. So we have 5 microliter bed volume and a 25 microliter bed volume. Depending on the amount of material you need and how fast you need to get those results, you would choose between those. You also have different cartridge chemistries, which give you a lot of ability to address different workflows, different needs. So you have those 2 options of sort of the flexibility. You also can use your own mobile phase for your chemistry. So if you have a cartridge, let's say a protein A cartridge, and you're purifying antibody out of cell culture supernatant, when you first start using the cartridge, you may need to have a matrix of wash conditions to get your best results, your cleanest sample. So you can do that all in parallel. So you can do experiment by design experiments on the AssayMAP Bravo, but then you may need to switch to a case where you're actually trying to address throughput. So you already have the experiment optimized as far as the chemistry goes, and then you can just use the same instrument to go through your samples. So the robot doesn't care what is in that liquid, so that gives you a lot of flexibility as well. The user interface, and I've talked about this a bit, there's a lot of flexibility there. And I'll talk about it more as we move forward. The last thing is just an example of the flexibility of the user interface and the kinds of improvements we constantly do. So with your system, when you get it, you will be able, the system keeps evolving. So we keep adding chemistry content, so new cartridge types, but we also improve the software on a regular basis. And one of the newest things that we're doing to address the software is making it 21 CFR Part 11 enabled so that you can work in both a non-compliant and a compliant environment depending on what's appropriate. And, importantly, if you're working in a non-compliant environment, you can hand off your assay to a compliant environment, and they can use the same system with leveraging the software features that allow enablement of compliance. So I had mentioned that and I've gone through the software being simple to use, and this is shown here too, but I want to also emphasize that it's easy to learn, not just for one application, for all the applications, because if you look at different examples of the applications here, you see they have the exact same look and feel. So once you've learned one, it's very easy to transition onto another application you may need for a different workflow. Also, these application methods are saved as electronic files, so it's easy to transfer. You can transfer to a different person, a different lab, a different geography, and not have the kind of concerns that one has when you're transferring SOP and people's personal preferences start to creep into the transfer. So another level of flexibility is the number of pre-configured, optimized applications that come with the workbench, which is our software portfolio. So in this slide, I'm going to show you icons of the applications. The name of the applications will be on the bottom, and the cartridge types that work with that applications will be on the right side. So this is the affinity purification application. And it's basically a fairly straightforward bind, wash, elute application. And we have cartridges with protein A, protein G, and streptavidin that can be used with this application. We also have the in-solution digestion application. You'll note that there are no cartridges associated with this icon because it doesn't use cartridges. It's a liquid transfer application. We realize for complete workflows, you're going to need both applications that use cartridges and ones that don't use cartridges. And I should mention the AssayMAP head can accept cartridges or pipette tips. So in this case, in solution digestion, you bring your sample plate to the deck, and you can do up to 4 sequential additions. And each addition of liquid, you can control the mixing, you control the volume, you can control the incubation time, the incubation temperature, and if the plate is lidded or not lidded. So if you have light sensitive samples, you can address that as well. Peptide cleanup, so cleaning your peptides up after, for instance, a digestion. We have 2 reverse phase cartridges that can be used with this application. C18 and RPS was a bare polystyrene based cartridge. And we have the phosphopeptide enrichment applications, which uses iron NTA and titanium dioxide. We have peptide fractionation. So you can take your peptides after you've done your digestion and fractionate them to go deeper into the proteome coverage. For this, we have strong cation exchange and the reverse phase resin. This may give you a hint of why we have two reverse phase resins is that this reverse phase resin is actually stable to high pH. Therefore, you can do this fractionation protocol to use as your first dimension in a 2D LC type of experiment. We have a reverse phase cartridge for protein cleanup, has much wider pores. We have the on-cartridge reaction application, which is a bit more complicated, but if after you immobilize an antibody, for instance, with the affinity purification application, you can draw liquid and enzyme into the cartridge so you can enzymatically treat the immobilized substrate, and then you can separate the soluble and immobilized reaction products from that reaction. And this can be done at elevated temperatures as well. And then finally, and another example is this, in this case, would be deglycosylation. So you could deglycosylate your proteins and collect the glycans and the deglycosylated protein. And the glycans, you can use this other applications for N-glycan sample prep and various dyes that we have available. So finally, what I'd like to turn to next is utilities. So I've talked about these applications, but we also have utilities. And by utilities, I mean, these are liquid handling operations. So again, utilizing the ability of the AssayMAP Bravo to pick up and use pipette tips, because we realize to build complete workflows, you really need a mixture of applications for cartridges and you need utilities for liquid handling. So these are some examples of the utilities. So we have reagent transfer, basically making daughter plates. Reagent aliquot, where you can go from a common source and fill various columns of the other plates. Normalization, where you can have a plate with various concentration of sample and then normalize it to a constant concentration and volume. Serial dilution and reformatting. Reformat is basically cherry picking. So with all these pieces comes sort of the ultimate flexibility, is combining them in different ways to address different workflows. So the very simple ones on top here, where you can do an enrichment of a protein, in this case an antibody, elute it, and you can do these sort of standard things you do with intact mass, protein quant, light and heavy chain mass. You can do more complicated by adding more pieces more steps onto the process. You can enrich, you can do, in this case, fabricator. So you are cutting the antibody on cartridge, and then you're collecting the Fab and the Fab and the FC region, and looking at analytical downstream processes. You can do one of the workflow that's a very significant workhorse workflow for proteomics and antibody characterization would be to purify the antibody, digest it, clean it up, and then look at these downstream analytical endpoints, peptide mapping, peptide quant, post-translational modifications. So again, all these pieces are available on the AssayMAP Bravo to put together to address these workflows. And lastly, as an example, certainly not the only examples, would be phosphopeptide enrichment, where you're digesting a cell lysate, you're cleaning up the peptides, and then you're enriching on something like an iron NTA. So you're looking at the phosphoproteome. So I hope I showed is that this tremendous flexibility, but still ease of use. So you can address a lot of things. The system keeps evolving, it keeps improving. And one of the ways it's improving is through the large capacity cartridges. So I'd like to turn to those next. So this is the two cartridge types that we have, size rather. So we have the standard capacity, is a 5 microliter bed volume, and a 25 microliter bed volume. Again, you'll notice that these are the same length. So they can be used in the system very easily. They interface with the probes of the syringes the same way, and they interface with the plates the same way. So you can seamlessly use these one or the other depending on your experimental need with no need to do any sort of hardware changes. Now, there are a little bit of software that has to be changed. And this only thing that needs to change in the software between the large and standard size cartridges is the default settings. So when you first open an application, in this case, affinity purification, what you'll see is that all of the values are blank. So, but what you can do is you can toggle to using this function here, navigate to pre-configured default settings for the different size cartridges. So depending on the cartridge you want to use, you would click on one of these. When you do, all the defaults will auto-populate. Then you can, if you have to change anything, you can change it. And then you can save that method and name it whatever you want so that you can use it again later or transfer it to another person. So very seamless, very easy. You can, that flexibility in itself is great, but it also has to be coupled with ease of use. So it's very easy to go between these two different cartridge types. Just to give you a sense of where we are on this, as far as launching the large capacity cartridges, because they are new, we have already launched and are available protein A and streptavidin. We are currently working on the C18 and RPS cartridges, which will launch in the near future. And the other cartridge types shown on here will be transitioned into large capacity cartridge, and the priority of those launches will be depending on what our users need. So any feedback will be most welcome, but they all are coming in the large capacity format. So that begs the question, why did we develop these large capacity cartridges? Well, like everything else we developed, it was driven by user need. And what we were hearing is that a lot of times people needed more capacity. Here's an example with a protein A cartridge. So you have cell culture supernatant and you need to purify an antibody to characterize. Often what you need to do is it has several different endpoints for that characterization. So you need enough sample that you can split into different workflows. So in this case, the top row here, we see that we've taken some of the sample, digested it, cleaned it up, and we're able to do these analytical endpoints. Again, this can all be done in the AssayMAP Bravo. But we also need some of the sample to go straight into other types of assays, some analytical, some functional, to fully characterize these antibodies. So how much capacity are we getting with these large capacity cartridges? So this graph shows the binding to the large capacity PAW cartridges. So on the x-axis, we have the masses that were loaded onto these cartridges. On the y-axis, we have the amount that was eluted off these cartridges. And the dotted line is the identity line, which represents if it was perfect correlation between loading and elution masses. And what you see is you get extremely good behavior up to the point where you start to hit the capacity limit, then you come off of this identity line. So for the 25 microliter cartridges, we can get about 800 micrograms of quantitative binding. And by quantitative binding, I mean less than 10% in the flow through of what you loaded. This compares to the 5 microliter cartridges where the maximum quantitative binding capacity was 100. So you can choose which cartridge fits your need. And the question might be, at first blush, why do you need so much material? And the answer is really that a lot of times when you do this characterization, you have mass spec type of analysis, which you need very little material, but you also have non-mass spec type of analysis where you need a lot more material. So another thing that you can, difference between the two cartridge types is time to results. So if we do an experiment where we're loading different masses of antibody onto, in this case, a 5 microliter cartridge, looking at the amount eluted, and again, the identity line is shown here, for a relatively slow flow rate at 2 microliters a minute, the behavior is near ideal, where ideal would be on this identity line until you hit capacity, then flatlining. And I told you this many times, this is a chromatography system. So what you would expect is as you increase the flow rate, you would fall off this identity line faster and faster because you're using up part of the binding capacity of the cartridge. So people often ask me, how fast is the flow rate should you load? And I would say that depends entirely on how much of the cartridge capacity you're 100 micrograms of the standard cartridge, and you should be between 2 and 5 microliters a minute. If you're using 20 micrograms of the binding, 20 micrograms of load, then you can do a much faster load rate. So if you then translate this onto a 25 microliter cartridge, you see the same sort of relationship, but you see at higher mass loads, you can go much faster. Like at 500, you can still go 20 microliters a minute or more. So choosing between 5 and 25 is really a twofold process. What capacity you need and what kind of flow rates do you want to have so to control your time to results. So one of the key features of the AssayMAP Bravo is low elution volume, which is shown here for the 5 microliter cartridge, where you see the total elution can be eluted off a cartridge in about 12 microliters. And then the other thing you see from this graph is you see the behavior you'd expect from chromatography. So I should back up and say that these are 2 microliter sequential elutions where you've loaded 50 microliters of antibody onto a protein A cartridge, and this is the amount of antibody in each elution. So you see what you expect from chromatography, a bell-shaped curve. But you also see a void volume in here. So the 3 takeaways: very low total elution volume, chromatography behavior, and void volume. And it should be noted that the software allows you to throw away some of this first amount of volume so you can even more completely minimize the amount that you have your sample, the volume you have your sample in. If we look at the 25 microliters cartridge, as expected, since this elution volume is related to the bed volume, the volume is higher, but it's actually still very low. Whereas if you throw away some void volume and you do this, you're talking somewhere in a range of 30 microliters, so extremely low volume. Now, if we look at reproducibility and recovery, this is the kind of thing that is obviously critically important. So in this experiment, the 5 microliter cartridge, we loaded 24 samples with various mass loads, and all the data is shown here with the sample number and the amount of antibody that was eluted on the y-axis. You see very good recovery and very low CVs. Again, the same sort of situation with the 25 microliter cartridges where you load various mass loads and you look at the elution and flow through, and this is the amount in the elution. You get very good recovery and very low percent CVs. So no matter which cartridge type you get, you're going to get very good performance. Finally, I'd like to show an example of the new cartridges that are coming out, the kind of workflow you would use. And this is an example where you would take some input material, digest it, and then clean it up. And then you would potentially go into a couple different paths. One would be an enrichment path where you're using something like your phosphopeptide enrichment to enrich for phosphopeptides, so the TiO2 or the iron NTA. And the other path might be an amino capture path where you're, again, isolating something out of this complex mixture. And then downstream, you'd have your analytical path. Now, should be noted that the large capacity cartridges would be important here because you're cleaning up a large amount of material to go into these enrichment steps, but also here where you could use it to multiplex in this amino capture step. So I'd like to end by saying that the AssayMAP Bravo platform really gives you great reproducibility, which is the absolute key. It has increased walkaway time, so you can set this up, press go, and come back and get your purified samples. It's easy to use. That's a very critical thing for us to make sure that the ease of use is there so people can very quickly get up and running, and it takes very little time to train and to use the system to get value out. And finally, it addresses a wide range of sample prep needs. By doing this, you can get the most out of your capital investment, and it keeps evolving to address even more and more needs. So I'd finally like to end by thanking the various people who have contributed to the development of this system over the years. Thank you.